Okay, so I think I should start now. Okay, so good morning, everyone. And so today I will tell you about some recent results of us. So I've changed the title of the talk to from evolution in changing world to something a little bit more technical to genetic diversity in changing environments. And most of the work that I'm going to talk to you about today is a part of the PhD thesis of Sachin Kaushik, and I hope many of you visited his poster on this topic. So some of this work is already published in Genetics this year or last year, and more hopefully be out soon. Okay, so I think now you all have the required PopGen background to understand what I'm saying. So today I won't show any equations or anything, and just speak qualitatively. And of course, if you have a question, just raise hand and ask. Okay. Okay, so we have seen this before last week, and several people have spoken about this. So this is this Lewontin's paradox, just to recall. So as you know that, as we discussed already, that if I assume the only neutral theory, okay, and we're looking at various measures of genetic diversity, specifically neutral genetic variation, which is the most common one, then the formula is that it should be equal to 4 times n times mu being the nucleotide mutation rate, and n being the census size. So therefore, the nucleotide the pairwise diversity should lie somewhere in this band. But the data is way off. This is pairwise diversity as a function of the census population size, and there has been a lot of discussion about how to explain it. Okay, and a lot of work has been done in that direction, and we discussed a few of these things last week, namely demography. That means the population size does not remain constant, it changes with time. If that is taken care of, some things can be explained. Another factor was selection at linked sites because of background selection, selective sweeps, etc. And nice unified account of these various ways that happens is given here, and I recommend strongly to have a look at this review. Okay, and there are other factors and so on. Okay, so I'm not claiming that we can explain Lewontin's paradox or anything. We are just doing our little bit to understand this, how genetic diversity is determined, how it may change. Okay, so one of the factors that we thought is the following: that most of the previous work assumes that the environment, the selective environment specifically, remains constant. Of course, there are exceptions, but most of the work assumes that the population is initially well adapted to a particular selective environment, okay, and then the environment is changed immediately, okay, and then in this new environment, typically the population will be maladapted. It will not be well adapted to that, and then the adaptation process will begin, assuming that once you have changed the environment, it remains unchanged thereafter. So that's the sort of the typical thing that is assumed in theory, and of course also you can do it in the lab. And it's not like a wrong assumption. It's a very reasonable assumption, provided the environmental change happens at a scale which is much slower than the evolutionary time scale. So if evolution is taking 1 million years, and environment is changing even longer time scales, of course, then you don't expect the environmental change to be of much importance. But of course, environment changes on all kinds of time scales. Of daily fluctuations in temperature, for example, seasonal variations, which changes temperature. Then there are changes over longer time scales, like over decades, for example, global warming, because of which temperature is basically changing linearly with time. Okay, so therefore one would like to understand how these changes in the selective environment can impact the evolutionary dynamics. Okay. So from now onwards, there is a sort of a broader meaning of environment. It's not just selective environment. It could be also demography. So environment, in general means selection as well as population size changes. Okay, so therefore one could ask questions about how changing environment due to changing selection or changing population size or both, how does that affect the evolutionary dynamics. Okay, so we have already spoken about demographic effects. So this is a question that people have studied for a very long time, and this was in the 1950s, 1960s. At that time, selection was not most of the focus was on neutral evolution. So I guess because of that reason, demographic effects, which is about population size only, and that's the only thing that enters when you are thinking of neutral evolution. That's the only parameter that enters. So this demographic effects have been well studied without selection for a very long time. Okay, but with selection, it's hard, analytically, and the studies are not that many, and people have started to think about selection and demography relatively recently. So selection may be changing or maybe not changing, but selection and demography is relatively new thing that people have started to think about. Changing selection, especially fluctuating meaning, the kind of thing I said about daily fluctuation in temperature. So that also people have considered it. And there's a book by Gillespie. There's an entire chapter focused on fluctuating selection. So people have again considered the effect of changing selection, but on genetic diversity is somewhat less studied. And again, more recently only, people have started to think about how selection change would affect the nitrogenetic diversity. And some recent works, but they do not consider demography. So then there's some obvious questions. What is the joint effect of changing selection and population size on mutual genetic diversity? We don't know. Similarly, what is the effect of changing environment at linked sites? So many of these works that I'm quoting above, they assume that there's no linkage. With linked sites, I'm not aware of any work actually that takes care of changing environment. So anybody out there who is aware of any such work, I would appreciate if you can let me know. Okay, so these are the sort of two broad questions that I have in mind, and I will tell you a little bit about our results on these two. Okay. Okay. So any questions so far? Okay.
Kavita, I have a quick question. Maybe you'll clarify this later. If so, I'll wait. But the second question, it's not, I guess, clear to me how environment changes will affect things at linked sites, or what do you expect, I guess. So if you're going to clarify this later, then I'll just wait.
Yeah, I'll just come to that.
Okay, thanks.
Yeah. So first of all, environment can change in many, many ways, right? So we are considering a particular kind simply because things are more doable there, and it's something people have not simply considered as much. So this is when the environment is changing periodically. So from school, you remember periodic variations, like a simple pendulum problem and so on. So all of you know what are periodic things, right? Okay, so we are assuming that both selection and population size are changing periodically with time. So let me first consider the changing selection part. So selection can change because of, for example, seasonal variations in plants. So what we're thinking of is a plant population, and there's a single locus of importance. And perhaps at that locus, the mutant is favorable in summer and it's not favorable in the winter. Okay, so it's that kind of effect of seasonal variation on mutant fitness is what we are trying to model here. Okay, so that's this up and down thing about some average. So we can consider several different scenarios within that. So for instance, this mutant may be, for example, this mutant could be beneficial all through. That means it is always beneficial. It's just that in summer it's slightly more beneficial than in winter, but it's beneficial in all seasons. So that's this scenario. Similarly, it's deleterious, the mutation is deleterious in all seasons. Okay. Or perhaps more interestingly, half or part of the season, it's beneficial, the mutation is beneficial in part of the season, part of the year, and part of the year, it's not beneficial, it's deleterious. So one can consider all three scenarios, and we have considered all three of them, but it turns out this is the most interesting one, in which the population encounters, the mutant encounters both beneficial and deleterious environments. Okay, so that's what I'm going to focus on. And that's one parameter in the model. Okay, but I'll focus only on this one. Other thing that you have to think about is how fast the environment is changing. So for example, the dotted line is an example of slow variation in the environment, whereas the darker one is when the variation is faster. So environment is fast or slow, that's another variable that you have to think about how that affects things. Okay. So this is a sort of model that we're thinking about the selective changes, the change in the selective environment. We also consider the demographic change. And example of that is the usual prey-predator dynamics. So all of you, I'm sure, have read or heard about it. So for example, this is the population of shoe hare and lynx. So as you know, then when the, let's say here, the hare's population is high, or rather the population of the lynx is high, and that of shoe hare is also high, but at some point, because predator population number is large, that of prey would start to decrease, right? And because there are too many predators. But then there's no food, so therefore predators population will also decrease. And now there are less predators, prey's population will also start to increase back. And then the cycle continues. So roughly, periodically, it is changing. I mean, so what's the period? There's 2 or 3, or there's chaos. And so this is a very interesting problem. So that's not what I'm thinking about, but it's a very simple model just to say how demographic changes could be periodic in nature. They could be oscillating. Of course, they have to oscillate about a positive number because population size is a positive number. Okay. So this is a sort of seasonal variation, the selective change and population size change is what we have in mind. Okay. And as I said already, I will focus only on the case in which the population encounters both beneficial and deleterious environments. I will also focus only on that case in which the environment is changing slowly. So let me elaborate this point. So what are the timescales that we have in this problem? Let's go for simplicity again to the static case. So environment is not changing. So if we don't have recurrent mutations, then we have these two timescales, namely due to drift and due to selection. So there's a time scale roughly 1 by S on which selection acts, and there's a time scale proportional to the population size or which drift acts. And a relative comparison of that gave rise to this product NS, which is what we discussed. Whether NS is large, then we have strong selection. NS is small, then we have weak selection. So those are the two time scales when we are in a constant environment. Now we have an additional parameter, which is the rate or the speed at which the environment is changing. So if the environment is changing very rapidly, faster is changing, that kind of thing is very well studied. And the answer is pretty simple. What has been shown analytically, numerically, and so on is that if environmental change is the fastest change, okay, so there are 3 scales, so you have to sort of order it. If environmental change is the fastest, then population does not track all the changes, it just sees the average environment. So this is a standard result in the subject. So if fluctuations are very large, it will not see these gradients here. It will just behave as though it's in a neutral static environment. Okay, so that's one reason. I mean, so of course, again, we have studied this also in our settings, which I will say later, but it's not a simple answer and it's not super interesting, so I will not discuss that today. Okay, what I will consider, however, is the opposite limit in which the environment is changing very slowly. In fact, the one in which the environmental time scale is the slowest amongst all these 3 time scales. And why would I do that? Because, if environment is changing fast, it's changing violently, you expect that you will see large changes in phenotype, you will see large changes in genetic diversity and so on. So it's kind of obvious what your changes you will see. But if environment is changing slowly, the effects produced may be subtle, they may be small, I mean, they will be small by continuity, but it's difficult to know that environment is changing or not. So it's kind of, it can produce subtle effects. So that's why I want to focus on the slowly changing environment. Secondly, I want to compare the answers to static case. So for a meaningful comparison, I would again consider slowly changing case. So because you cannot compare Usain Bolt to a standing person, right? So it doesn't really make sense. So therefore, I will focus therefore for the slowly changing environment. Okay, so that's the sort of things that I'm going to focus only on today. But we have studied all the parameters and so on, and you can look up the papers or talk to us later. The simple one-line thing that I'm going to find is that even in slowly changing environments, there are changes, and from what you expect from the static case, and they happen when we have deleterious mutations as well as beneficial mutations. So the impact comes due to deleterious mutations, which at least I was very surprised to see. Okay, so that's a sort of a simple summary. And now let me describe to you what we expect. But again, before that, since we have seen stochastic trajectories last week quite a lot, I just want to show you what happens when environment is changing. How do these stochastic trajectories look? Because it's a little difficult to know. Allele frequency is changing, right? Because of selection, because of drift, and on top of that, selection is also changing, right? So here's an example of these trajectories.
Kavita,
Yeah.
I have a question.
Yeah.
So in your previous slide, when the environment is changing rapidly, the population is behaving as if it's facing an average environment, right?
That's correct.
So over a longer time scale, won't it look like that the population is facing a static environment?
Yeah, it's facing a static environment with an average, with this average.
Yeah.
Average of the environment. So if the population, if it's a beneficial mutant always, it will see as though the selection coefficient is 0.04.
Okay.
Yeah. Okay. So that's the standard result. Yeah. Okay. So that's why it's sort of not very interesting. And actually, Gillespie's book, I think, has mostly this kind of discussion. Okay. Okay. So this is the one in which we have the trajectories when selection is changing. But you can see a slight difference because it's a very slowly changing environment. And if you didn't tell me the trajectories, they look more or less same to me as though they are in a static environment, right? That's what you expect intuitively, right? Okay, so fine. So this is how it looks. But if the environment is changing at some sort of intermediate rate, then there is something more which is visible at the allele frequency level. And let me just explain that bit. So what you have here is that the selection is positive till here. So there are two lines. The red one is for selection, and this is for the allele frequency, the labels. So till over here, the selection is positive, and as a result, the allele frequency increases because selection is positive. But then selection becomes negative. So in tandem, the allele frequency also decreases. And then it goes on for a while, and at some point, the examples that I'm showing you, it so happened that allele frequency could, get rid of genetic drift, and it is no longer so sensitive to changes in selection, and it fixed. Okay, so here I could still guess that perhaps the selection is changing. Here I certainly can't. Okay, so it's still interesting, however, to see that how the slowly changing environments could affect things. So that's what I want to focus on. Okay. So I'll first describe one problem, which is about selective sweeps. And I'll assume that there's no demography because we did not study it. And I'll assume only the selection is changing in the periodic fashion, like I described to you just now. So quickly, let's recall, we have seen this last week. So we have these bunch of individuals, a lot of loci. These black ones are all neutral variations. So there are some with circles, some with this minus sign. And at some point, a variant arises. Maybe it is deleterious, maybe it is beneficial. And if it happens not to get lost due to drift, then it will sweep through the population. And if there is sufficient linkage, then it drags along this neutral variant with it. So now you have a loss of linked genetic variation. So that's something which we have seen already. What I want you to remember are just these couple of points, that the time that played an important role here was this fixation time. the time it takes to go for this new mutant, this de novo mutation, to sweep through the population. So this is the T sweep or the fixation time. Those who understand these things, this is a conditional mean fixation time. Okay, so that's what's being compared with the recombination time. If fixation time is very long, there's hardly any loss of genetic diversity. If fixation time is rather short, there's a loss of genetic diversity. We also discussed this symmetry, the Maruyama-Kimura symmetry, where I said that this fixation time for a positively selected allele, for a positively selected allele is same as that for the negatively selected allele, but that was assuming that the mutant is co-dominant. There's no dominance, but the symmetry is actually a bit more general than that. So let me explain that. So suppose we have a single locus and we have this diploid population, and this homozygote has a fitness 1, and the other homozygote, the mutant homozygote, has a fitness 1 plus S. Then the heterozygote's fitness, the contribution to that is a fraction H from the mutant homozygote and 1 minus H contribution from the wild type. Okay, so that's how we can compose the fitness of the heterozygote. If H is 1/2, that means both of them contribute equally, then we have codominance. If H is not equal to half, then we have dominance. If H is larger than half, then we would say that the mutant is dominant, otherwise it's recessive. Okay, so this symmetry is a bit more general. It says that you should compare a positively selected mutant which is dominant with a negatively selected recessive mutant. So suppose H is 0.8, that means a dominant mutant. So this will become 0.2, right? So this is dominant, this is recessive. Similarly, this is 0.2, this is 0.8. So when this is recessive, this is dominant. Okay, so it's saying to compare positively selected dominant with negatively selected recessive, or positively selected recessive with negatively selected dominant. Okay, yeah, Amir?
I'm just curious why you're writing 1 plus HS as this expanded form. I didn't get that.
Why am I writing what 1 plus S?
1 plus HS for the heterozygote. It will be 1 plus HS, no? This entire expression.
Yes. So
why are you writing it in this form?
Yeah, let me repeat. Yeah. So I'm saying that is because this is a fraction, for the heterozygote, a fraction H contribution comes from the fitness of this homozygote, and rest, which is 1 minus H fraction. So it's 1 minus H into 1, right? So H is the fraction which is the contribution coming from 1 plus S, that is this homozygote, and 1 minus H from this one. Does it help?
All right. Yes. All right. Yes.
Okay. Yeah. Okay. So that's all. So you're right. It's 1 plus HS, but I wanted to tell you why is it 1 plus HS in fact. Okay. So this is the symmetry, which is more general. Okay. And the consequence of the symmetry was simply that because, as we learned, the fixation times are connected to diversity, and therefore deleterious sweeps and beneficial sweeps, they will give us the same level of neutral diversity. Same level in the sense of the heterozygosity, for example. Okay, so this is what I want you to just keep in mind. And now we are going to change the environment and ask perhaps right now the most exciting question, which is what happens to the symmetry, right? Before I do that, let's get familiar a little bit more about this changing environment, because it took us a long time, somewhat long time, to just, get our head around this, that, this is changing allele frequency as well as selection is changing. It's a little complicated. So let me just explain this point a little bit more. So what I'm plotting here is the selection coefficient as a function of time. Okay. First of all, you have to understand that in a changing environment, a mutant is not beneficial or deleterious, because environment is changing. Today, it's beneficial in summer, this mutant, but in winter, it is deleterious, right? So I cannot label it as a beneficial or a deleterious mutant. I can, of course, say, what is it what's its sign when it just comes into the picture, when it just arises. So for instance, it could be initially beneficial, it could be initially deleterious. Okay, so that's one thing to remember. So for example, if I have a beneficial mutant, it could, because positive selection, so it could either come along this part, which is the first quadrant, okay, or it could come like this. In this case, it's coming, the beneficial mutant is coming in improving environment, selection is increasing. If it happens to arise here, it's coming in deteriorating environment. Okay, fine. To understand, to see what Maruyama-Kimura symmetry does in changing selection, what do we have to do? We have to compare S and minus S, right? So which means we have to take the mirror image of this part about 0. So suppose I have this environment, I have this mutant, which is positively selected, and it came in an improving environment. What is the minus of this? The negative S of this, which means it will be a curve like this, no? Right? So the mutant will come over here, and it will be in a deteriorating environment, which is what is plotted here. Okay? So I have to compare to understand Maruyama-Kimura symmetry in a changing environment. I have to compare an initially beneficial mutant in improving environment with an initially deleterious one in deteriorating environment. Similarly, you can compare beneficial in deteriorating with deleterious with improving environment. And then dominant recessive, we have to take care of, as I explained to you just now. Okay, so slightly more involved about what you want to compare when you are thinking of changing environments. Okay, so now I'll show you the data from our numerical simulations. So this is what is shown. Let me explain. So what we have, this simulation is for a 2-locus model only. So what we have is a first locus, which is neutral locus, which means the wild type as well as the mutant are neutral. And then there's a second locus in which the wild type is neutral. And the mutant, however, has this changing selection coefficient. Okay. And then if the sweep occurs, there is a change in the neutral heterozygosity of the first locus, which is the neutral locus. That's the genetic variation which we are tracking. So which is what is plotted here as a function of the rate at which the environment is changing. Okay, so this is the classic Maynard-Smith and Hague model. Okay, and the difference simply being selection is now changing with time. Okay, so it's just the classical stuff with this small difference. Okay, and the deleterious is the recessive one, and beneficial is correspondingly dominant one. And here's the result. So if environment was static, this green and red points would have coincided. Okay, static is over here, static limit is over here. But when environment is changing very slowly, when it's changing very slowly, these points don't coincide anymore. Okay, so first thing that we learn is when we choose appropriately the mutants to compare with, that there's no more Maruyama-Kimura symmetry. Frankly, we are not surprised. It's not surprising. So if this is based on this diffusion theory, this understanding of this symmetry, and if you are familiar with those equations, you expect that this symmetry will no longer hold. So it's interesting that there's no symmetry anymore, but at least to us, it was not surprising. Okay, but what is interesting here is that you see the deleterious sweeps. So remember, deleterious sweeps in quotes. I cannot call something as a deleterious mutant, right? So these deleterious sweeps are more affected compared to the beneficial ones. So the canonical value would have been 0.26, 0.27. This goes max 0.24, 0.23, whereas this goes all the way till 0.4 or so. Okay, so that's the second point. The deleterious sweeps are more affected than the beneficial sweeps. And in the inset, we have the data only for slowly changing environment, where again, the same point is made more strongly. The beneficial data hardly changes when we change only over a order of magnitude, the rate of environmental change. It hardly changes, whereas the deleterious one has changed quite a bit. So even a slowly changing environment, deleterious sweeps are more affected and beneficial ones are not. And by the way, when you have a slowly varying environment, it is okay to think of deleterious as deleterious sweeps, because environment is changing so slowly that if you start out as a deleterious, you'll finish your job while environment is still deleterious. Typically, that's true. Okay, fine. So we would like to understand what's going on here. So like Deepa was asking, what's the expectation? So I thought, yeah, it would be interesting because, there is so much study on beneficial sweeps, and all of that literature assumes that environment is not changing. So if we put environmental change, are there more sweeps? Is there a more reduction, less reduction? What happens? So we did not have any expectation, but at least I expected that the beneficial sweeps would be affected. So it was very surprising to me that it's the beneficial ones which did not change much. It's the deleterious ones which did. So I'll try to explain in some simple qualitative way why that is so. Okay. We can look at this two locus model, but it's pretty hard. So again, we generally work in the diffusion theory, and it's almost impossible to make progress with it when we have more than one locus. So here's one strategy that physicists or people. Yes, Amir.
So on the y-axis, I didn't understand what the relation with heterozygosity is. I thought it was mean fixation time in the Mario. So how?
Yeah, that's because I said that here that the fixation time and diversity are related.
Right. All right. Okay.
So it's from there.
Okay.
Okay. Yeah. So we can look at interdiversity, but analytical is very hard. Okay, especially if I have to deal with deleterious mutations where I cannot neglect drift. If you can neglect drift, as you can when you're looking at beneficial sweeps in static environments, it's possible to make a lot of progress. Okay, but when you have deleterious sweeps, it's just almost impossible. So here is what I was saying, that when you do analytical work, it's not like this is the model that I have to study. I have to worry only about that quantity. You can look at something which is doable. So that's one strategy which physicists and other people who do analytical work employ. So in our case, we found it easier to work with the fixation time. We wanted to look at the equations fixation time because they are tractable. And like as Amir said, asked, what's the relationship? This is something we did last week. The fixation times and the diversity are related to each other. Okay, higher the fixation time, lower the diversity, and vice versa. So therefore, we decided to look at a fixation time. And because I want to compare beneficial and deleterious, and I told you, okay, so I told you there are 3 time scales. I'm going to look at a slowly changing one, but then there are other 2 time scales also, the drift scale and the selection scale. So therefore, you have strong selection and weak selection. Strong selection is a reasonable thing only for beneficial mutations, because deleterious mutations, if there's strong selection, they simply will not sweep, they simply will not fix. Okay, so we have studied strong selection for beneficial mutations, but I will not present those results because I want to focus on the effect of deleterious sweeps. So therefore, I will consider the case of moderate selection. Yes, go ahead. I can't see your names.
I just want to make sure I'm understanding correctly what is going to be addressed here. So you're saying the mean fixation time of the neutral variation associated with a deleterious allele, correct? That's what you're going to examine as a way to address how much the neutral diversity floats around or doesn't float around.
Yeah.
Okay. And the second question I had is with in the previous plots as well, you were talking about, again, heterozygosity associated with either a deleterious or a beneficial allele. And I guess in this model, you are assuming that the selection coefficients are similar for the beneficial and the deleterious allele, and yet you see this sort of asymmetry that is being uncovered.
Yeah, they are, everything is same, right, except we are starting with different initial condition. This versus this.
Okay, great. Thank you.
Yeah. Was there one more question? No. Okay. Yeah. So let's go back. Okay, so therefore I will just focus only on moderate selection and slowly changing environment, simply because I want to look at deleterious and beneficial together. Okay, so let me explain this. So we have gone from heterozygosity to mean fixation time, but keep in mind they are related. And again, I'm not showing the data, it's in the paper. You can put those 2 figures together and you will see qualitatively is the same behavior. Okay, And here, however, I'm showing you not the results from numerical simulations, rather these are coming from this diffusion theory. Okay, so let me explain this because there's a lot of stuff on it. So what is shown here is how fixation time changes with a function of dominance. When I have fixed the amount of selection, I have also fixed the rate at which environment is changing. The blue points correspond to the case when the environment is not changing. It's a static case. So if environment is not changing, this is how this fixation time would change with dominance. And we can understand that. But let me focus on just these 2 points first. First point is the deleterious sweeps are more affected than beneficial ones. What does it mean? So the blue points are the static one. If I have a beneficial mutant, that means it came initially beneficial, right? So this mutant, its fixation time is not very different from static one. But the mutant which was initially deleterious, its fixation time is substantially different, and that too when it's particularly recessive. Okay, so 2000 versus like 2200, 300, 400, whatever. Okay, so we want to understand these 2 points. So there are other things, but let me just focus on these 2 points. This one, let me first understand. So let me explain this, maybe using the Jamboard, simply because I find it easier to explain. Visible, Jamboard?
Yeah.
Okay. Okay, so the question is simply this. So why deleterious sweeps more affected? Okay, so let's go back to something simple. The static case first. Remember, we spoke about fixation probabilities. You heard that in Michael Lynch's talk also yesterday. So when you have a beneficial mutant, then its fixation probability, P-fix, is approximately twice the selective advantage. This is for the codominant mutant, okay? Whereas the same thing for a deleterious mutant is something like n times the selective effect. These two are very different functions. In the static case, if you look at these formulas and if you just plot them, and I strongly encourage you, and I think Sachin did this exercise in the tutorial. For a beneficial mutant, if you change the selection a little bit, your chances of fixation are not, they don't change much. That's the part about this function. But if you have a deleterious mutant, you change S a little, fixation probability will change quite a lot. It's a very fast changing function. So that is the really key point. And that's why the quantitative theories are very important. These formulas are very important to understand these little things. Okay, so this is the key point. For beneficial mutant, the fixation probability changes only, it's less sensitive to changes in the selection. Whereas for deleterious, it's more sensitive. So what has that to do with what I want to say? So suppose I have a static environment and there are some trajectories, okay? 2 of them went to fixation, let's say, and 1 got lost. And we are interested in this. So this is our static one. So I will calculate fixation time using these 2 guys, right? So now if I have a initially beneficial mutant, selection is changing, but the chance, because this is slowly changing environment, selection is changing slowly, the chance that these two don't make it is negligible. Most likely, these trajectories are pretty similar to the static case. In the deleterious case, however, because a small change in selection makes a large difference, it's possible that this got lost, but maybe all 3 got lost. Because the chance that you make it, and therefore, you were calculating averages from here, right? So that average will be affected. The sort of trajectories which are going to contribute to your fixation time, they're very different set compared to the trajectories that contributed for the static case. Okay, so that's really the key point here. Okay, so this is the basic point. Okay, so that is all. I I can find the slide. Okay, so that's what I wanted to say. Small difference in S makes a large difference in the fixation probabilities, but not the beneficial ones. And it's because of this sort of thing. Okay, so this is a basic one. And you can calculate these things even in a changing environment. Okay, and that's basically the reason why these numbers, the red numbers, are different from the blue numbers. Okay, why recessives? We thought of a simple argument, but I think I'm no longer convinced of it. At least I'm not sure today. Okay, but again, sort of similar thing happens, and this is if we calculate the fixation probability with dominance, it turns out again that the effect of changing environment is more for recessive mutants than the dominant mutants. And then the same argument that I just gave you can be used to understand why there is a more difference for the recessives than for the dominants. So this you can find in the work of a former PhD student, Archana, who is now a postdoc at Arizona State. Okay, so you can look that up. Okay, so it's basically that. So if you guys can take this little idea about how this difference makes, how that translates into other things, such as what I'm showing you here, I think I have done a good job. Okay, yeah. Go ahead, Deepa.
So do you expect that something similar should also happen regardless of what is the rate of change of environment? I mean, it should, right? Like what you're saying is like a fundamental difference between how sensitive the deleterious and beneficial mutations fixation times are. Two changes in both N and S.
Yes.
I mean, N, right?
So if you change environment faster, that changes will be more. Right, so there will be larger changes, but it's just that it's kind of obvious that there will be larger changes, but it's just these limits are more subtle because they're tiny changes, and yeah, so it's more interesting. Of course, there will be larger changes, sure. Yeah.
Okay, thanks.
Okay, good. And so it's not necessary that the fixation time only increases, and therefore only heterozygosity only increases. It works other way around also, depending on where the arrival time of mutants. Again, those who work in stochastic processes, let me just tell you the equations that are going under this. 1 second, Deepa. Those are time in homogeneous processes. As a result of that, arrival time matters. So I hope you understand, but otherwise you can look it up. Yeah. Okay, so that is all. So that is a simple basic thing that I just wanted to highlight here and stress here, which is that changing selection, you expect that it can impact the diversity levels. And what we find rather surprising to us it's the deleterious ones, which are more affected. And the way we explain it is basically how these formulas, the very basic Kimura formulas, how they are related. What is the form of those formulas for beneficial versus deleterious? Now, deleterious sweeps are hardly studied. Okay, simply because one says that deleterious mutations they're hardly fixed. Sweeps are about fixation. So why study? But on the other hand, deleterious mutations are much more common. So, but somehow deleterious sweeps are not well studied. And I'm also not sure whether, what we are saying here about importance of deleterious sweeps, whether it matters in the bigger picture. I'm not sure, at least not today. Yes, Deepa.
So I guess this is really interesting and has nice implications for people trying to find the genetic basis of either adaptation or, allele frequency changes for deleterious cases as well. So what you're suggesting then is that for deleterious alleles, they should behave more like their selection coefficient based on the selection coefficient that you expect, right? Whereas with beneficial alleles, it should be that even if there have been minor fluctuations in the selective environment, you should still be able to use the sweep pattern to find beneficial mutations. Is that fair summary?
Yeah. That's correct. Yeah. So that's basically what I'm saying here. So for deleterious ones, so that's the last line here, which is the beneficial. Okay, let me just go through my script, which is that, like I said, that deleterious sweeps are less studied, and I'm not, as I said, we need to know more to judge whether or not deleterious sweeps really matter. Okay. one question therefore one can ask is, so this study, because we did not expect deleterious sweeps to be important, so therefore we were surprised. And but what the study tells us is that deleterious ones are more affected, and therefore maybe we should look at this problem in the context of background selection. So what is background selection? Remember, these are deleterious mutations. They are less likely to fix, but very likely to get lost. And that's what background selection is about. So because deleterious mutations are more sensitive to the changes in the environment, we expect that, I think, background selection would have a more important effect. It will have more important effect in the context of background selection. But that's something we have not studied, and that's suggests next project. But like Deepa, you said correctly, what this study also tells us, so you might think of it as a negative result, that, oh, nothing is happening if environment is changing for the beneficial mutations. It's actually a good thing. There's a huge amount of literature, a lot of work has been done for beneficial sweeps. And what our work suggests is that you don't have to go and redo all that stuff. With changing environments, at least slowly changing environments, beneficial sweeps, I mean, you're safe, your beneficial sweeps work is robust, it's not going to change much. But if you have deleterious stuff, you better watch. Okay, so that's basically the simple summary. Okay, so I have about 10 minutes or so. And since we spoke about SFS last week a little bit, I thought I'll just tell you a little bit about what we are thinking about in that along that direction. So this model that I just want to tell you now, it's different from the previous one. There's no linkage anymore. In the first problem, I neglected demography, but now I will actually consider selection and demography. So this is a sort of more general model that people have considered so far. But selection and population size, both are going to change periodically. But let's start with simple basic stuff. What is SFS? So we saw that a little bit, but I thought I'd do just a little example here. So we have here 6 individuals, and we have 8 loci, and at each locus, there are some variants. So let's say 0, somehow I know it's an ancestral allele, and 1 is a derived allele or the mutant allele. So because there's no linkage, there is evolution happening independent at each locus. So what one does is, suppose you give me the sample, okay, of these 6 individuals and these 8 loci, what the variation is. So what I do is just to count how many mutants are there. There's 1 here, 2 at a second locus, 3, and so on. Okay, so I count the number of mutants at each locus, at each site, and then make a histogram of that. Just 1 second, Deepa. So what I do is then I ask that how many sites have a single mutation. So there are 1, 2, 3, 4 of them. So that's with 4. How many sites have 4 mutations? None, which is that here. That's how we construct SFS. Yes, Deepa.
No, sorry, that was my earlier hand, which was not.
Oh, sorry. Okay, fine. Okay, so this is the SFS, and that's how you construct from your data. But how do we understand, like, what should I expect, what the shape and so on is like? So this is standard classical model called the infinite sites model, which I sort of said last week, but let me just explain this a little bit. So what one is thinking about is a very long sequence. There are lots and lots of sites, okay? And for simplicity, I'm just taking 3 samples, okay? So let's say at the first locus, there's a mutation happens, and nothing changes in other ones. And after some time, this locus is doing its own thing. That is the mutant frequency. It will fluctuate because of drift. Maybe there's a selection or not. So these numbers will fluctuate, right? In the meanwhile, perhaps new mutants came at the second and third locus and so on. And if after some time, these mutants decided to get lost, whereas this decided to get fixed. And similarly, some mutant appeared at the 5th locus and it also got lost and so on. For SFS, we look at polymorphic loci. So these ones where fixations or losses happen, we don't care. We don't want that data. We want to look at only polymorphic loci. So that's the sort of data that I showed you here. They were all polymorphic. Okay, and then from that, SFS is constructed. Okay, but this assumes 2 things. There's no back mutation. Once 0 has become 1, there's no going back. And that has to do with the fact that we are considering a very long sequence. So hitting the same site twice, that's unlikely. Secondly, there's no linkage. So therefore, this model is applicable to sexual populations, the sort of thing that I showed you in the context of Lewontin's paradox. Okay, and so then you can work out from the constant environment, the expectations are the following. The SFS for the neutral environment looks like this. It just changes, it just decreases. For deleterious mutations also, it decreases, but the magnitude is very small. Okay, so it's not 1 over here, it's much more small. But the shape is basically that of a decreasing function. For positive selection, it's qualitatively different. It's a more like a U-shaped pattern, asymmetric U. So those who like, collect data and interested in making contact with biology and want to analyze this data, I explained to you how to collect this data and how to do things with it. But it's not just data collection and so on. There's actually deeper stochastic stuff going on, which I did not explain. And I'll just mention it in one line, that in just like in the first part, we had this fixation time, conditional mean fixation time, which played an important role. SFS is proportional to what is called sojourn time. It's a time that an allele frequency spends in a tiny strip along X, centered about X, frequency X. So in X minus dx and X plus dx, how much time it spends, okay? So we didn't discuss it. So for those who are interested in stochastic processes, here is this question, and which is related to SFS, which people measure all the time these days. Okay, so what happens in changing environment? So I'll just tell you one answer. So here, in the blue, I hope you can see, in the blue is shown the answer, like I said, change monotonically decreasing answer for SFS for neutral case. And the green open are the ones for changing in for selective one, which I just showed you, U-shaped versus decreasing one. In the fast changing environment, like I said, you see just the average environment. And therefore the red points for the fast, they behave like the neutral one. But in the slow one, it behaves, all the environment is changing. It behaves like as though there is selection. We are considering environments which are neutral on an average. But the SFS comes out to be U-shaped function. Okay, and there's a simple understanding of that. But I think I'm running out of time. So let me just skip that and just show you that SFS is a lot of information. But again, you can look at heterozygosity, for example, and if selection and population size were constant, you will get this black curve. If you have demography as well as changing selective environment, this is what you get. Genetic diversity reduces. It can reduce even further if you look at even more general model. So the selection and demography, they don't have to go hand in hand. They could be changing with different frequencies. They could have a phase difference. And in some cases that we studied, the answer is very different from what you see. There's no structure compared to what you have in a constant environment. Okay, fine. So I think I should just stop here now. So static environment is great. I mean, just to think about it is much easier, and it's a reasonable assumption, at least at some short time scales. But like I said, for example, if you're looking at long time scales, when there's global warming, you cannot expect such, if you are, you cannot expect that environment is remaining constant, and therefore you have to take this into account. And it produces different effects. This, of course, has been realized a long time ago. It's not a new thing that I'm saying here. But I think what we are saying somewhat new is that it's a deleterious mutation. So 2 of our studies, second study, I did not tell you the reason, but it turns out it's again deleterious mutations which make this difference. These differences are coming again because of deleterious mutations. Okay, so, and that's because we are very sensitive. Okay, so the difference, however, is coming when we have deleterious mutation into the picture. And like I said, that one could ask, for example, do these results connect to Lewontin's paradox and so on? I think we have to study a bit more general model. So, for example, the selection linked site model, at linked sites model, it has some subtle effects, which things we did not expect, and sort of different from what we would have generally thought. But it's just a two locus model, and there are other assumptions. So that's something which we are planning to now look at it a more general model and see if we can quantitatively connect with the Lewontin's paradox. Okay, so I think that is all I want to say. Thank you. Unless there are questions, otherwise we can stop. Yeah.
This is super interesting. I don't understand everything yet, but okay. But it's scary that the side frequency spectrum changes so much for neutral diversity. With a little bit of a change in the environment. I'm trying to wrap my head though around how do I think of the time scale? So, for example, compared to the mutation rate or something, what is the time scale of the environment and environmental change? Like when you say slow, like how slow is slow, like in terms of generation time or something?
Right. So mutation rate is sort of a finite quantity mean. So you have to think of it as composite parameters, right? So n mu is fixed.
Okay.
What is changing here is ns could be large, ns could be small, but all those so timescales are like n 1 by s and environmental change. So here I'm assuming the environmental timescale is much longer than the timescales over which drift happens or selection happens.
But drift happens and selection happens, I mean, for the complete fixation of something or complete loss of something.
No, it's just
or even
order.
partial. I see.
Of that order. If you double the n, double the time takes.
Okay.
So it's just of order. I mean, that's sort of the mathematical concept perhaps, but that is of the same order. Yeah.
Okay, thanks.
Yeah. Go ahead, I can see the names. Ravi, can you speak? I can't hear you. Ravi, I can't hear you. Maybe if you can write the question in your chat. Okay. Any more questions? Okay, fine. So we will meet, I guess, at 2 o'clock. Thank you.